So we're going to pick up where we left off talking about eukaryotes, the domain eukarya. We're going to recap what they are, and we're going to talk more about when they first appeared in the fossil record and how they evolved. So how did we get eukaryotes? How did they pop onto the scene? So when we're talking about eukaryotes, they're one of the three major domains, the other two major domains being the bacteria and the archaea. And what we've done is basically take these three domains and blown up the eukaryotes to show all the major sort of eukaryotic lineages here. And this is the common ancestor of all eukaryotes. And you can see a couple of the major eukaryotic groups like fungi, so things like mushrooms, like animals, and like land plants. So these are typical eukaryotic groups that we think of, although there's lots of other major lineages that you can see here. And we talked about the traits that define the eukaryote. So they can be uni or multicellular, and we talked about the fact that complex multicellularity is a trait that's unique to the eukaryotes. They can range in size from tiny unicellular organisms all the way up to the largest organisms that we know on Earth. They have in general a larger cell size. They tend to be 10 times bigger or larger than prokaryotic cells. They have membrane-bound DNA that's wrapped up in a nucleus, and they also have organelles, so little things floating around inside the cells that are membrane-bound, like mitochondria, chloroplasts, and the other organelles that you guys have probably learned about from high school. They have linear chromosomes inside their nucleus. So linear chromosomes are a trait that's unique to the eukaryotes. And they can reproduce both asexually by mitosis and sexually by meiosis, and the ability to reproduce sexually is unique to the eukaryotes. So, first question, where should linear chromosomes be placed on this tree? A, B, C, D, or E? If you said C, you are correct. So, linear chromosomes is a trait, it's a synapomorphy, it's a unique trait that is derived and defines this entire group, the eukaryote. And the eukaryotes themselves are a very diverse lineage. In fact, we currently know about 7 superkingdoms that are eukaryotes. Okay, so when I talk about superkingdoms, you can think about something like Animalia being a kingdom. So remember, it goes domain, kingdom, phylum, class, family, order, genus, species, and the major taxonomic groups. And you can have something that's a little bit bigger than a kingdom, but a little bit smaller than a domain, and that would be a superkingdom. And you can see that there's 1, 2, 3, 4, 5, 6, 7 major superkingdoms that we know of. And so I wouldn't expect you to memorize all of these different lineages, all of these different major kingdoms, or any of the superkingdoms either. But what I do want you to be able to understand is that there's a lot of diversity in terms of the eukaryotes that exist that are out there. And I would also want you to be able to read this tree in general. And also just sort of understand like things like animals and fungi are over here. So these are major kingdoms. Let's see here. You can see the green algae and the land plants are here. Red algae and brown algae are here. Things like dinoflagellates are here. So the rest of these things are sort of protozoans that you're probably not super familiar with. But you should have a general idea of what's going on in this tree, and especially just be able to read it in terms of interpreting a phylogenetic tree. So what do I mean by that? If we're looking at this tree, to which group below are fungi most closely related? So remember, fungi are right here on the tree. Okay, if you said F, A, and C equally, so fungi are equally related to animals and choanoflagellates, because fungi share a common ancestor here with animals and choanoflagellates. So fungi are equally related or equally distantly related to animals and choanoflagellates. They are less closely related to these dictyostellid slime molds and way less closely related to the land plants, because the common ancestor that it shares with those groups is all the way back here. So when did eukaryotes first appear? So remember this figure that we've been looking at of the sort of ancient history and the origins of Earth and the major evolutionary events that occurred on Earth. When did the first eukaryotes appear according to this figure? Okay, if you said 2 billion years ago, you're correct. So we see the first single-celled eukaryotes popping onto the scene around 2 billion years ago. And once again, if we compress all of this onto a 30-day calendar, this 4 billion years 4.5 billion years onto a 30-day calendar. It's not until about day 20 that we see eukaryotes coming onto the scene. So it's pretty late in the game that eukaryotes evolve. So why did it take so long for the first eukaryotes to appear or to evolve? So if you think about it, just putting it into perspective, about 4.5 billion years ago, that's when the Earth formed and there was no life on Earth. And it took about 1 billion years later for the first prokaryotic cells to appear. So the first prokaryotic organisms, the first life to appear. And then you had diversification within the prokaryotes. And it wasn't until about 1.75 billion years after the first prokaryotic cell came around that we saw the first eukaryotic cells appearing. So what happened here? What two important traits gave rise to or had to evolve to give rise to eukaryotic cells? And two very important traits that I want to discuss are the nucleus and the mitochondria. So mitochondria and nucleus, these are two traits or adaptations that are unique to eukaryotes and eukaryotic cells. Okay, so clicker question. Answer these questions 1 and 2. All right, if you said C, that only the eukaryotes have a nucleus and only the eukaryotes have mitochondria, you would be correct. So these are unique traits. Remember, mitochondria and organelle, and only eukaryotes have an organelles, and a nucleus is a unique trait that defines the eukaryotes. So where did these unique traits come from? Given that only eukaryotes have these traits, how did these traits evolve and give rise to the eukaryotes? So when we talk about the evolution of a nucleus and a nuclear envelope, one of the running hypotheses is that essentially you had an ancient ancestor of a eukaryote that had its chromosomes floating around in the cell. And of course it has a plasma membrane surrounding that cell. So we're talking about a sort of unicellular ancestor that has these chromosomes. And then there were infoldings of the plasma membrane, and so you see these infoldings occurring. And eventually those infoldings sort of moved in and pinched off and became what's called the endoplasmic reticulum. And they also formed a full sort of membrane around the nucleus, or the nuclear envelope. And so we have these infolding membranes pinching off, forming a nucleus, a nuclear envelope, and something called the endoplasmic reticulum, which is essentially just a series of membranes that serve as an area to sort of process proteins and RNA that are passed out from the nucleus, okay? And this would be the sort of origin of a eukaryotic cell with a nucleus. So why would these infoldings evolve in a prokaryotic cell? Well, one hypothesis is that this has to do with increasing surface area to volume ratios. So if you can think of a cell over here, C, and that cell gets more and more infoldings as you go from C to B to A. That's kind of like this example that we used with Lori, where when she's standing like this, she's covering up this area of her surface area, but when she's standing like this, she has her armpits and her inner thighs exposed, and so that means that she's artificially increasing her surface area while her volume is staying the same. She's increasing her surface area, and so are the cells in this. Okay, so if cells can infold, they can increase the volume. If cells can infold, they can increase volume without decreasing surface area to volume ratios. So they can get more and more volume, but they can also have more and more surface area to volume ratios, so they can get bigger without decreasing their surface area to volume ratio. And so the idea here is these infoldings allowed eukaryotic cells to get bigger and also make space for all those sort of organelles and potentially a nucleus and things like that. So let's think about these different cells, whether you're talking about cell A, B, or C, where the black line represents the plasma membrane. So where are these cells taking in food and letting out wastes? Think about that for a minute. So if you said through the cell membrane, you would be right. So they have to get nutrients in from the outside environment through the plasma membrane, and also have to pump waste out through that cell membrane to get them out of the cell and out into the environment. Okay? So given that, which of the following is true? If you said cell C will lose waste more quickly per unit volume than cell A, you are correct. So cell B does not have a lower surface area to volume ratio than cell A. Cell A would have the lowest surface area to volume ratio because if these have the same volume, B has more infolding, so it's going to have more surface area relative to its volume. and cell B will absorb nutrients more quickly per unit volume than cell C. That's false because cell C is going to have the highest surface area to volume ratio, so it's going to be absorbing nutrients more quickly per unit volume than cell B will. And so the idea here is that selection or evolution by natural selection should lead to the formation of a nuclear envelope basically by, over time, selecting for cells that have more infoldings, because in theory, those cells, they should have a higher surface area to volume ratio and overall higher relative rates per unit volume of waste excretion and nutrient absorption. And so there's selection for more infoldings over time. And that selection over time led to the evolution of the nucleus. So the idea here is, this is once again just one hypothesis for how the nucleus formed, is that you have selection for more infoldings, and eventually those infoldings formed the nucleus. So we've talked about one hypothesis for how the nucleus formed. Let's talk about hypotheses for where mitochondria came from. And that specific hypothesis is something called endosymbiosis theory. So endosymbiosis roughly translates to inside together living. In symbiosis, the definition of it is individuals of two different species that live in physical contact with one another. So this isopod that lives inside, it's a parasite that lives inside the mouth of a fish, and it basically latches on to the fish's tongue and lives inside there. This would be an example of symbiosis, where two different species, the isopod and the fish, are living in physical contact with one another. And endosymbiosis is when one organism of one species lives actually inside the cells of another species. Okay, so this endosymbiosis theory first came about from this woman, Lynn Margulis, who basically came up with this theory and hypothesized that mitochondria actually originated when a bacterial cell took up residence inside of another cell type around 2 billion years ago. And so there was a lot of controversy surrounding this theory. People were sort of up in arms about it, saying that's a crazy idea, that mitochondria might have evolved through this endosymbiosis, or of a bacteria actually moving in and living inside of another cell of a different species. So let's look at what the actual evidence is for this idea. So first, let's understand endosymbiosis. Essentially, you had some host cell that was sort of a precursor to eukaryotic cell, and you had a bacterium that could do aerobic cellular respiration. And so what happened was that bacterium got engulfed by this host cell, but it didn't get lysed, so it didn't get digested. Instead, it sort of took up residence inside of that host cell. And what the bacterium supplied, because it could do aerobic cellular respiration, supplies lots and lots of ATP to that host cell. And remember, ATP is sort of the currency, the dollar currency of the cell. It's the chemical currency of the cell. There's a lot of value to having an organism that can produce a lot of ATP, and aerobic cellular respiration can produce a lot of ATP. And so that bacterium supplies that can do aerobic cellular respiration provides a lot of ATP to this host cell. And in exchange, that host cell provides some protection and also provides some of the precursors that that bacterial cell needs to do aerobic cellular respiration, okay? So answer this question. If you said B, aerobic cellular respiration generates more ATP than other metabolic processes, that would be correct, okay? So remember, the big benefit that this host cell is getting is because this bacterium can do a metabolic process that the host cell was not able to do originally, and this metabolic process produces way more ATP than this host cell could do, and so it's getting a lot more energy from that bacterium. And so the idea here is this happened somewhere deep in our evolutionary past, and then this gave rise to a cell that had a selective advantage, and then that host cell would pass on that endosymbiotic bacteria that became the mitochondria, that is the mitochondria, onto its offspring. And that gave rise to eukaryotic organisms, or at least organisms that had this endosymbiotic bacteria that allowed them to do aerobic cellular respiration. And so the idea is that this happened somewhere back in the deep evolutionary past of the eukaryotes. So this is a trait the evolution of mitochondria through endosymbiosis that got passed on down the phylogenetic tree and defines the eukaryotes. So they inherit it from a common ancestor. So that's a great story, but what's the actual evidence for this? So one piece of evidence for mitochondria, and let's just go ahead and actually answer this question to start. If mitochondria are once bacteria, what should their DNA look like? If you said their own circular chromosomes, you are correct. So if mitochondria were originally bacteria, they should have their own circular chromosomes, just like bacteria have circular chromosomes. And in fact, mitochondria all have their own circular chromosomes. So this is a great piece of evidence in support for endosymbiosis. Okay, what other pieces of evidence do we have? When we look at mitochondria, they in fact have two cell membranes. And that is exactly what the theory of endosymbiosis would predict. Because if an original host cell initially engulfed a bacterium, it should wrap that bacterium in a little piece of its cell membrane that it pinches off. And so if you have this sort of active phagocytosis, you should have this right here is the precursor to mitochondria. This is a bacterial cell. It should have its own cell membrane, and then it should have another cell membrane wrapped around it that it got from its host cell that engulfed it. And in fact, that's exactly what we see, that if we look closely at mitochondria, they have two cell membranes. One here that you see outlined in blue, and then a second one that you see outlined in purple, which is exactly what the theory of endosymbiosis would predict. So that outer membrane from pinching off of the host cell, and an inner membrane from the original bacteria. And in fact, now that trait's been passed on and on and on from different mitochondria. So other evidence for endosymbiosis, it turns out mitochondria replicate by binary fission, just like other prokaryotes do. So if you remember back the way that prokaryotes reproduce, they do it through this process called binary fission. And that's exactly what mitochondria do as well. So they have a circular chromosome, they make copies of that circular chromosome, then they pinch off and split in half. And they actually do this reproduction or copying of each other, this binary fission, within host cells completely independent of reproduction of the eukaryotic cell. So the eukaryotic cell does its reproduction to make copies of itself, and the whole time completely independently of that, these mitochondria inside of it can reproduce independently by binary fission. Okay, so given all of these pieces of evidence, which of the following support the theory of endosymbiosis? If you said B and D, you are correct. So remember, mitochondria have circular chromosomes just like prokaryotes and just like the theory of endosymbiosis would predict. If mitochondria came from a bacterium, they should also have circular chromosomes. Mitochondria have two cell membranes, so you see the sort of pinching off of the two cell membranes. You see that here as well. Mitochondria sexually reproduce by meiosis. No, that is a process that's unique to eukaryotes, so we should see mitochondria, if they did come from prokaryotes or a bacterium, they should reproduce by binary fission, which is exactly what we see, and they do have their own separate chromosomes. And so B and D are the two correct answers that support this theory. So, how did eukaryotes evolve from prokaryotic organisms? We've talked about the nucleus, we've talked about the mitochondria. What about chloroplasts? So let's talk for 1 second about chloroplasts. So which of the following groups, bacteria, archaea, and eukarya, have chloroplasts? If you said C, you are correct. Remember, chloroplasts are organelles, and so they're unique to the eukaryotes. And so if we're going to put chloroplasts on this tree, we might put them somewhere here, although not all eukaryotes have chloroplasts, only some eukarya, not all of them. And if you look at the proposed origin of chloroplasts, they're also proposed to evolve from endosymbiosis. And so the idea here is that somewhere in the deep evolutionary past of eukaryotic organisms, there was a sort of initial eukaryote that had nucleus, it had mitochondria, and some of those went on to become sort of typical animal cells that only have mitochondria to produce ATP. But you also had some cells that, along with having mitochondria, had a second round of endosymbiosis, where essentially you had this photosynthetic prokaryote. So remember, mitochondria are from a prokaryote that could do aerobic cellular respiration. And then photosynthesis, so chloroplasts can do photosynthesis, and this is an organelle that lives inside eukaryotic cells. Those potentially came from engulfing a photosynthetic prokaryote, and so this became the organelle of a chloroplast. And if you look at present-day plant cells or photosynthetic eukaryotic cells, you can see that those photosynthetic cells have chloroplasts, so this is one organelle that can do photosynthesis to produce sugars, and then they also have mitochondria as well, and those mitochondria can use some of those sugars that the chloroplasts produce to convert those sugars into ATP. So if you look at photosynthetic eukaryotic cells, they have chloroplasts and mitochondria, just like this theory predicts. And when you look at chloroplasts, they have all the same bacteria-like characteristics as mitochondria. So they have circular chromosomes, they have their own genome, they have a double cell wall, and chloroplasts also replicate by fission. And so all of these pieces of information support this theory that chloroplasts also evolved through this endosymbiosis of a bacterial species that could do photosynthesis. So, last clicker question. Which of the following groups contain circular DNA in their genome? If you said both, you are correct. So remember, prokaryotes have circular DNA, but also remember that a eukaryotic cell has both linear DNA, which is the sort of major nuclear DNA that forms up the majority of its genome. However, it also has circular DNA through chloroplasts and or mitochondria that it has in its cells. And these chloroplasts and mitochondria, remember, have their own independent genomes and replicate independently by binary fission.